We did our project on RNA sequence of lung cancer.
So some background information on lung cancer is that it affects the alveoli, which is responsible for absorbing oxygen in your blood from inhaled air and removes carbon dioxide from the blood when you exhale. Common cells are lung squamous cell carcinoma, which is found in 30 to 50% of lung cancer cases. There are two types of lung cancer: non-small cell lung cancer, which is the most common, and small cell lung cancer. Lung cancer is the most common type of cancer in the world.
Some background information on tobacco smoking is that it could lead to cancer of the lungs. It can also lead to coronary heart disease and stroke and chronic obstructive pulmonary disease. And smokers are at high risk for stroke, blindness, deafness, back pains, and osteoporosis, as well as some other diseases. And a lot of chemicals are found in tobacco smoke, over 7,000, which are all harmful for the body. And one of these includes nicotine, which is the addictive drug that causes people to have the urge of smoking, as well as you can find ammonia and carbon monoxide in it.
Okay, so our hypothesis was that RNA sequence will help us understand the molecular inquiry in response to tobacco smoke, smoke exposure, and lung cancer pathogenesis by identifying gene expression differences not integrated or accurately measured by microarrays.
Some of our methods that we conducted were that we took 8 genomes from 8 individuals and split them up into 2 different analyses. So we basically took half of those patients, so 4 each, and we took the RNA sequence of them and compared them to smokers and who have lung cancers and smokers who don't, as well as healthy individuals who smoke and healthy individuals who don't smoke. And we characterize the impact of smoking and lung cancer on the airways transcriptome using RNA sequence. So here's a histograph that shows pretty much summarizes the p-values of all the genomes that were collected for the first 4 patients. Most of the genomes don't show significance or any variation, except the very few that at the bottom left end that do show significance. Because only a few genomes were significant in the analysis, the top 2 that are significant can be seen in this volcano graph. And the name of the most significant genome is the FOSB proto-oncogene, and it's located in the gallbladder, bone marrow, thyroid, and in the lungs, as well as 27 other tissues.
In the next 4 set of patients, we did an analysis where there was 3 experimentals, which were smokers, 1 without lung cancer, 1 healthy, and 1 with lung cancer, and then the control was a healthy non-smoker. You see up top, the blue one is the most significant because the higher up the points are, the more significant or more expressed the gene is compared to the control. And then the green ones that fall at the bottom represent the ones that have no significance or no difference between the control and the experimental. In the second analysis, we ran 3 controls and 1 experimental. The 3 controls were people without lung cancer in comparison with the 1 individual who had lung cancer. Again, the most significant is all the way up top, which was the same gene present in the other analysis. When comparing the 2 genes that were the most significant that were found in both analyses, the one on the left was the one present all the way up top. Some of its functions include chemotic activity towards neutrophils, cell signaling, and also antiviral activity during a viral infection. It's found in salivary glands, spleens, lungs, and 22 other tissues in the human body. On the right-hand side is another gene where its functions are a little bit different. It has anti-porter activity, transmembrane transport activity, and it's also found in the ovaries, endometrium, and 25 other tissues, also including the lungs. But the one on the left is more significant just because it's found more often in the lungs compared to the gene found on the right. So observing the one on the left would be more beneficial in further research.